So I'll introduce you. So next up we have Courtney Harrison. She received her BS in Animal and Nutritional Sciences from West Virginia University in 2018. After graduation, she worked as a research intern at the Conservation Fund Freshwater Institute, where she assisted in the design and performance of research focused on environmental water quality involving recirculating aquaculture systems used for growing Atlantic salmon. Her interest in animal health led her to Michigan State University Aquatic Animal Health Laboratory in 2019. Courtney is interested in the effects that infectious diseases have on the health and survival of juvenile lake whitefish and lake whitefish recruitment in the Great Lakes. So Courtney, if you want to start your slideshow, you can take it over from here.
Can everyone see this?
Yes, we can. We can see the PowerPoint if you want to go into the present mode.
Yeah, that's where I'm trying to. Oh, okay.
There we go.
All right. Thanks again for the introduction. Good afternoon. My name is Courtney Harrison. I'd like to take a moment to thank the organizers for having this great idea to have us all come together and have an opportunity to talk about some research I find particularly exciting involving fish health. I'm thrilled for the opportunity to speak with you all today, even if it's from a safe distance. I'd also like to take a moment to thank the co-authors of this study, the current lab members of the Michigan State University Aquatic Animal Health Lab, who contributed to this study, and Travis Brendan and Mark Ebner for their research collaboration. So before I begin, I'd like to share a little bit about me. I'm from West Virginia, and I've spent most of my life in the Mountain State. I studied Animal and Nutritional Sciences at West Virginia University, where I graduated with a Bachelor of Science in 2018. After graduating, I was a research intern at the Conservation Fund Freshwater Institute and gained experience working with salmonids reared in recirculating aquaculture systems. And my interest in animal health did lead me to Michigan State University in January of 2019, where I'm in my second year as a master's student, and I have the pleasure of working with a great team of fish health enthusiasts while I perform research on a subject I'm very passionate about. So the picture you're seeing is a primary species in the research I'm part of. The lake whitefish, Coregonus clupeiformis, within the family Salmonidae, comprises the most valuable commercial fishery in the 4 upper Great Lakes, being valued at over $4.7 million per year in Michigan alone. So before I moved to Michigan, I never even heard of a lake whitefish. So now everywhere I go, I see signs advertising smoked lake whitefish, and I know it's this fish species, and I can't help but smile and get a little hungry. So not only does this lake whitefish support the Great Lakes economy, but it's invaluable to the lake's ecosystem. The lake whitefish is a bimvivore that feeds on zooplankton and other invertebrates, and they are also a primary vehicle in cycling energy from the lower trophic levels to the higher trophic levels within the lakes. So the chart you're seeing here now illustrates lake whitefish commercial yields over time. So like I said before, lake whitefish comprise an important commercial fishery. But as you can see in this graph, their numbers declined in the past. In the 50s, the introduction of sea lamprey to the Great Lakes devastated lake whitefish commercial yields. But with better management practices, their numbers recovered. Unfortunately, there have been recent declines in adult lake whitefish abundance and growth. Studies show that possible factors contributing to these losses could be from changes within the Great Lakes food web and invasive species, among others. And similarly, there have been a decline in early life stage recruitment. So what is recruitment? Simply put, recruitment is the introduction of new individuals to a population, meaning successful recruitment requires that eggs to be successfully laid, hatch, and for fry to survive to join the populations. So now that we're on the same page about recruitment, these declines in recruitment of young lake whitefish is concerning, and we don't know what's causing these declines. So what hasn't been investigated is the role of infectious diseases, and if they could be behind what's causing these declines. So there's been some research focusing on infectious diseases affecting Great Lakes lake whitefish, and the papers I'm showing you here come close to representing the totality of that work. These papers made solid contributions in clarifying some of the infections that occur in Great Lakes lake whitefish. But what's important to note is that these studies focus solely on adults. So now when you look at the microbial infections that were detected, you'll note that they all have the potential to be shed either in reproductive fluids or within infected eggs. So for example, Rhenibacterium salmoninarum has the potential to be transmitted in offspring of infected fish through intra-ovum transmission, at least in other salmonids. And similarly, Carnibacterium, Aeromonas salmonicida, and viral hemorrhagic septicemia can be readily shed with reproductive fluids in other fish species. However, whether this occurs in Great Lakes lake whitefish has never been explored, and this is an important knowledge gap considering recruitment of young fish is poor. And so there are actually several related knowledge gaps. So for example, can these pathogens be present in the reproductive tissues of adult lake whitefish? And if they are present in the reproductive tissues, do those same pathogens persist during spawning, and are they transmitted in the eggs along with reproductive tissues? And if they are transmitted with the reproductive fluids, does that lead to infection in the sac fry? And if they do become infected, will those same infections persist in juvenile and older stages of the fish? But above all else, are these infections affecting juvenile lake whitefish health? So to answer these questions, this overarching hypothesis was formulated. Fish pathogens that are transmitted from adult lake whitefish to their progeny act as bottlenecks to the recruitment of young fish to Great Lakes populations. And so to address this hypothesis, several objectives were formulated, but I'm going to focus on just two of those today. The first of which is to assess the disease status in wild spawning adult lake whitefish. So to address this objective, and based on historical data, spawning aggregations were collected from Lake Superior, Michigan, and Huron over a period of 2 years. Sites were selected to represent both good recruitment locations, shown by blue stars, and poor recruitment, shown by red stars, within each lake. So that means there are 2 paired sites in Lakes Michigan, Huron, and 1 site in Lake Superior. So I went out on commercial fishing boats and collected 60 lake whitefish from each location. So this was another first for me. I'd never been on a commercial fishing boat before, and it wasn't exactly like Deadliest Catch, but there was one morning I was in Whitefish Bay on Lake Superior catching lake whitefish, where the temperature was freezing and it started snowing halfway through our collection. And I was constantly getting my hands and arms soaked from freezing water, only for the captain to tell us that the heater in the main cabin wasn't working very well. So I tell my family and friends I had the true Northern Michigan experience that day. And after I collected 60 fish of varying sizes and of different sex, I kept them alive during transport back to our lab for a thorough pathological and diagnostic investigation. So upon arrival to our lab, fish were euthanized with an overdose of MS-222. Individual fish length and leaks were recorded, and we collected blood, notable internal and external lesions, and kidney, spleen, and heart tissue. We also specifically looked at testes and ovaries and took reproductive tissue samples from each fish, because remember, we're looking for that potential pathway for infected parents to infect their offspring. So this project took a multifaceted approach to recover and identify a multitude of pathogenic microbes, including, but not limited to, using an array of bacterial growth media that supports a diversity of bacterial fish pathogens, multiple cell lines to screen for several viruses, collecting blood for individual packed cell volume, and an enzyme-linked immunosorbent assay was used for the detection of Rhenibacterium salmoninarum. So at this time, I'd like to share with you some interesting results to date that I'm particularly excited about. So over the period of 2 years, we collected 60 fish per site per year, as I showed you previously on that map, which equated to sampling 600 adult Great Lakes lake whitefish. So kidney, spleen, and heart tissue was inoculated onto 3 different cell lines, including EPC, FHM, and CHSE. Cell lines were incubated at 15 degrees Celsius up to 4 weeks post-infection and observed for any cytopathic effect. So there was no observable cytopathic effect on any cell lines, so fish were considered negative for viral infections, at least for viruses able to replicate on these cell lines. So next I'll show you a multitude of bacteria and more significant findings to date. So I recovered several yellow pigmented bacterial isolates from kidney cultures on the flavobacterial selective medium TIES. After initial morphological and characterization and subsequent PCR analysis, I confirmed their identities as Flavobacterium cycrophyllum. And I was super excited to discover this because not only was this the first confirmed detection of Flavobacterium cycrophyllum in Great Lakes lake whitefish ever, but because of what I know about this bacterium and its effects in other fish species. So I'll talk more about the significance in a minute, but first I want to show you how prevalent infections were. So here I'm showing you the prevalence of Flavobacterium psychrophyllum based upon kidney culture results in spawning adult lake whitefish. So I'll be showing a few histograms in the following slides that will have a similar layout to this. The prevalence is on the y-axis as a percentage, and the collection locations are along the x-axis, and are separated by lake. Also, each location is denoted with a blue star or red star to represent good and poor recruitment, respectively. So as you can see, infection prevalence was relatively low, ranging from about 1.5 to 3.5% at 2 sites. And of note, infection intensities were also quite low, with only 1 to 2 CFU per fish per microliter of kidney tissue. So given that, what could these infections mean for Great Lakes lake whitefish? Well, Flavobacterium psychrophyllum is a devastating salmonid pathogen, causing substantial losses in the early life stages of trout and salmon raised in aquaculture and hatcheries around the world. In fact, one of the names for the disease this bacterium causes is rainbow trout fry syndrome, because of the substantial mortality it causes in early life stages. We also know that this is one of only 2 bacterial pathogens with strong evidence for intra-ova transmission. So coming back to lake whitefish, what can I say about the infections mean at this point? Well, given the low prevalence and intensity, it could be that this bacterium is not a big concern. However, it is important to note that when lake whitefish spawn, they migrate to shallow spawning grounds and congregate in large numbers. So in other words, they don't necessarily social distance during this process. So this begs the question, for there to be effects on juvenile lake whitefish health, would the prevalence need to be very high? So another serious pathogen known to be transmitted in the egg is Renibacterium salmoninarum. And yet again, we detected this bacterium in spawning phase Great Lakes lake whitefish. The ELISA assay we use to detect Renibacterium salmoninarum not only detects the presence of the pathogen, but the infection intensity as well. And this is shown by different colors noted as low, medium, and high. So let's take a look at Lake Michigan first. It's clear that within this lake, the site with poor recruitment, Bailey's Harbor, has a higher prevalence of Renibacterium salmoninarum and a good recruitment site, Menominee. So then we see in Lake Huron, the same thing is happening. The prevalence is higher in the poor recruitment site. However, the Lake Superior results are interesting, in that the prevalence is highest at this site, but also note that these were all low titer infections as estimated by ELISA. So just hold on to that thought and let's have a look at what we found when we look specifically at reproductive fluids and gonads. So we didn't find any in Lake Superior, which is interesting. How about when we compare by sites within lakes? Again, in Lake Michigan, they had a higher prevalence in the poor recruitment site. And this is the same story in Lake Huron. So I also want to mention that this is also important because this is the first detection of Renibacterium salmoninarum in reproductive tissues of Great Lakes lake whitefish, which is noteworthy given that Renibacterium salmoninarum is transmitted intra-ovum. So now we're beginning to see that potential pathway for parents to infect their offspring. However, we can't forget the health of the adults. In this study, we saw a subset of fish that had pretty serious signs of disease, like the fish you're seeing here. This fish had a medium titer of Renibacterium salmoninarum and had lesions such as severe ocular hemorrhage, diffuse hemorrhaging on the isthmus, and severe splenomegaly. So before we leave Renibacterium salmoninarum, what do we know about its effects in other Great Lakes salmonids? Well, it was a contributing factor in Chinook salmon mortalities in Lake Michigan in the late 1980s and early 1990s. It is the second of 2 bacterial fish pathogens with strong evidence of intra-ovum transmission, and it's still an issue in the Great Lakes. So the potential to negatively affect Great Lakes lake whitefish health is there. So another bacterium recovered from our initial investigation yielded this familiar morphology. And based on phenotypic and biochemical characterization, we presumptively identified these bacteria as belonging to the genus Carnobacterium. And so following molecular analysis, we could confirm the bacterium's identity as being Carnobacterium multiromaticum-like. So this is the Carnobacterium infection prevalence in adult lake whitefish in 2018. So doing site comparison by lake again, we see in Lake Michigan, the poor recruitment site has a higher infection prevalence. And similarly, we see this in Lake Huron. And also, it's noteworthy that a subset of the bacterial cultures we recovered, Carnobacterium, came from the reproductive tissues. So I find this very exciting, revealing yet again a potential pathway of the bacterium being shed within eggs while fish are spawning. And so a subset of fish collected that was later determined to be infected with Carnobacterium had serious signs of disease, such as severe hyperemia of the swim bladder, vasculature, and notable thickening of the swim bladder wall. When some of these swim bladders were opened, we discovered they had a mucoid turbid fluid within the swim bladder lumen, the consistency of which can be seen being aspirated by the transfer pipette. And also of note is the more pronounced opacity and thickening of the swim bladder wall, concurrent with diffuse hemorrhage. So if you remember the colony picture from earlier, this is the pure growth that came from this fluid collected in this picture. So again, for the first time, Carnobacterium was recovered from the reproductive tissues of spawning adult lake whitefish, which is significant given the bacterium has a penchant for reproductive tissues in other salmonids and causes post-spawning mortality. This is also a substantial finding given that the recruitment of juvenile lake whitefish is poor. So to summarize, we've made some significant discoveries, although there seems to be more questions than answers. We found 3 bacterial fish pathogens in spawning adult Great Lakes lake whitefish, 2 of which were found for the first time in reproductive tissues, and 2 had higher infection prevalence in poor recruitment sites than in good recruitment sites. So could these contribute to poor recruitment in juvenile lake whitefish? I can't answer that today, but it could be a potential pathway from parents to offspring. So now that we found some substantial bacterial pathogens in adults, will we see similar findings in the juveniles? So the collection process for juvenile lake whitefish was a little different from collecting the adults. So I went to sandy beaches that were adjacent to the spawning location of adult fish, and same for wild juvenile lake whitefish in the late spring and early summer. In some instances, we collected 150 lake whitefish from one site, and in another, we collected 24. And then there was that one day when I was only able to catch 1 lake whitefish after staining all day at multiple beaches. But fortunately, I collected 325 wild lake whitefish last year. And I say fortunately because it's not as easy as it sounds. Another student and I visited each site, did multiple trawls, and walked up and down the shorelines to look for juvenile lake whitefish, all while sifting through hundreds of other young fish. And after spending all day on the beaches, I brought the fish back to our lab and did thorough clinical, pathological, and diagnostic investigations. So just like the adults, there was no observable CPE on all cell lines. So they were all also negative for viral infections. And I also collected and cryopreserved over 100 bacterial isolates, which investigation and identification is still underway. So the next steps will include laboratory experimental challenges to determine what these infections mean. Juvenile Coregonus clupeiformis have never been experimentally challenged against microbial pathogens in a laboratory setting, and I've been rearing several hundred juvenile lake whitefish in our lab to be used for these challenges. So I'm pretty excited to begin these soon and have a better understanding of multiple pathogens' effects on the health and mortality of young lake whitefish. And once experimental challenges are complete and wrap everything together, data generated from the study could be used to better inform stock recruitment models, how infectious diseases could lead to uncertainties in these models. So although findings to date have generated more questions than answers, we've made several discoveries that are important, including recovering 3 bacterial fish pathogens, 2 which were recovered in the reproductive fluids of spawning adult lake whitefish, and are more common in poor recruitment sites than in good recruitment sites. I also recovered multiple bacteria from wild juveniles that I'm still identifying, and I'm really excited to determine what they are given that no one has ever bacteriologically examined wild juvenile Great Lakes lake whitefish before. But even with analyses ongoing, we've highlighted potential pathways for bacterial transmission from infected parent to offspring. And I'm super excited to begin to understand not only the potential links between infection statuses of spawning stocks and their offspring, but also what impact these infections have on juvenile lake whitefish health towards better understanding the potential contributor to their poor recruitment. So at this time, I'd like to acknowledge the Great Lakes Fishery Trust for funding this project, the fishing boat captains for letting me come onto their boats and collect adult lake whitefish, the MDNR Fisheries Division, Chris, Paul, and Ted for helping me beach seine for juvenile lake whitefish, the Little Traverse Bay Bands of Ottawa Indians for supplying me with juvenile lake whitefish for my experimental challenges, my good friends in the department for helping me with fish collection, my friends and colleagues at the MSU Aquatic Animal Health Lab, and my advisor, Dr. Tom Slock. And at this time, I can take any questions.
Thank you, Courtney. That was an excellent presentation. If you have a question for her, just go ahead and unmute yourself, or you can put it in the chat box. Okay, Courtney, we have a couple questions.
I'll ask a question.
Okay.
Great job, Courtney. So I wonder if that you were looking specifically for some pathogens, but I'm just wondering if you go back to the map of the sample sites, did you collect it, or do you know if water quality has been evaluated? And again, I'm from Costa Rica, so I have little to no understanding of the Great Lakes. So could you point out, if any of those areas are closer to human population, like large human population, where contamination of the water might be also higher in some of the areas, and that might be playing an important role in recruitment of the brood stocks as well. Because I know you're focusing on pathogens, but I guess a lot of it, water quality changes, climate change, and then of course human interaction might also play an important role.
Yeah, and those those are great. Those are great suggestions too. I mean, again, we are focusing mainly on infectious diseases for this study, but we do consider climate change to be a factor, especially stressing fish in their young early life stages. But what we haven't looked at in this particular study is the role of human populations in the surrounding area. So yeah, that's a great suggestion. So thank you.
Courtney, we have a couple questions in the chat box here. The first one from Adrian Lopez is wondering if you looked for the presence of parasites like microsporidians or mixosporidians that might be vertically transmitted as well.
So yeah, we did recover some parasites in especially the adults. I didn't find any in the juveniles. And a lot of them too, they were so small, it was hard to get a skin and gill biopsy from the juveniles. But in the adults, we did find some gyrodactylids and some cystodocula within the swim bladder lumen. So that was an interesting find.
And then one from Dave Groman. He was wondering if you did any histopathology on any of these fish.
Yes, we did. So if I can go back to one part of my presentation in particular, so this tumor, actually, this is the only tumor we found from the 600 adults that we collected, but it turned out to be a spindle cell neoplasm. But also other notable lesions, internal and external, were collected, and histopathological investigation is still underway. So I'm hoping to make some discoveries on that end very soon.
Excellent. And then one more here in the chat box right now from John Drennan. He's wondering if the Carnobacterium could cross-react with the Rhinobacterium ELISA, leading to a higher OD readings, resulting in positives for our cell. Does that make sense?
So I haven't really considered that, and I'm sorry, but I don't really know how to answer that, because this is something I haven't considered.
So, Courtney, I'll jump in. This is Tom Locke. John, excellent question. You know, back in the day, actually, early on because we rely upon our ELISA in the state fish hatcheries to screen for Renibacterium. And we also find, at least in some of our captive brood stocks, we were finding Carnobacterium infections. And so early on, we did test our ELISA, which is a derivation of the one in the Blue Book with some modifications. And we did not get any cross-reactivity between Carnobacterium and Renibacterium.
Sunil here. Excellent presentation, Courtney. So
Thank you.
I'm a virologist and molecular person, so I will be more biased on virology side. When I was working with Nick on the project, like part of the previous presentation, so we tried 3 fish cell line, 2 temperature, no success in virus isolation. But with the next gen sequencing, we have identified different viruses. So do you have any plan to try some of the sample for NGS also, or you just wanted to go with these selected bacteria only?
Yeah, no, we were just sticking with these cell lines that we've used, and this is coming from like previous literature and previous studies conducted on Great Lakes like Whitefish. So that's why we chose the cell lines that we use, EPC, FHM, and CHSC. Because right now, the only virus that has been found in adult Great Lakes like Whitefish has been viral hemorrhagic septicemia virus. So that was the main virus that we were, targeting, but again, like we're casting a wide net. And so any viruses that were able to grow on those 3 cell lines, there was no CPE on those.
But Sunil, I'm just going to jump in one more time. So we did also save those tissues, correct, Courtney, at -80.
Yes.
So that's something that we don't have the funds for in this study, but it is something that we very much are interested in.
Yeah, we can talk later, but yeah, we have improved our method significantly from this project. So I think we can get better results in next NGS run or so. So yeah, we can talk later about this. But
Okay,
it will be good
thank
to see the virome pattern also in these samples.
We have time for one more question if anybody has one.
Hey, Courtney, this is Jan Levy. Nice talk, thanks.
Thank you.
Just curious, how is recruitment measured in the lake whitefish? Do they collect young of the year samples, and how old are they when they're measuring recruitment?
So I'm not entirely sure how they're measured, but they do look at the juveniles, so the younger fish.
Sure.
So I assume that they would just come from like state and federal agencies that do these yearly recruitment models or recruitment collections of young lake whitefish.
Okay. Reason I'm asking is the incubation period. Can you comment on the incubation period for Renibacterium? Is it a longer incubation period in younger fish, or can you get acute disease in young fish with that?
So one of the reasons behind this study is we don't know the incubation period or the effects in Renibacterium salmoninarum in lake whitefish. I do know that it does have a long incubation period in other salmonids. So my best guess is that it's probably another long incubation period in this species, but again, we don't know that for certain.
Okay, thanks.
Mm-hmm.
All right, thanks again, Courtney and Sumesh, for the excellent presentations today, and everybody for joining us. Please come back next week. We have a double feature of vaccine development, so if you're interested in that topic, be sure to tune in. Virtual round of applause for the 2 speakers, and I hope everybody enjoys their weekend. Have a good day.
Thanks. You too.
Thanks.